Welcome you all to the course on bioelectrochemistry. So, at some point or other, you must have heard about electrochemistry. So, if you divide this word electrochemistry, so it is essentially two words which will emerge as a layman, right. One is electricity, the other one is chemistry. So, you can use electricity to break a molecule, which we call as electrolysis or electrolysis. Using electricity, lysis means breaking. So, you are breaking a molecule. Okay. This is one way you can look at the problem. The other way you can look at the problem, where because of a chemical reaction electricity is generated, which makes perfect sense if you think of it. What is a chemical reaction essentially? A chemical reaction is there is a transfer of electrons from one species to another one. And if you even try to make it very simple, if we look at different elements which are present on the floor of earth or maybe in the universe, each element sits at a unique potential to donate electron. So, you can have a hierarchy series saying that say A has the highest potential, it has the highest power, it can donate electron, then B which is slightly lesser than A, like C which is even lesser than B, likewise and so on and so forth. So, each element or each entity has a certain power to donate an electron, and similarly you can have the table the other way. Each entity has a certain unique strength to capture an electron. So, between two entities, the probability of a reaction taking place is essentially what is the electron donation power and what is the electron acceptance power. And what an electrochemist does, they quantify the amount of electrons which are getting transferred from say entity A to entity B or vice versa, right. And in that process, if the electron gets transferred say from A to B, what will happen? So, whenever we talk about the flow of current, we say current flows from higher potential to a lower potential, right. So, if the reaction takes place say between A to B, if the electron flows, so automatically A is sitting at a higher potential, B is sitting at a lower potential, the electron can flow, right. So, there will be change in a voltage. So, there will be change in the voltage between A and B. So, there will be a delta V change. So, we will measure that voltage. How we can measure that voltage? Now, this is one aspect. The other aspect is that how many electron flow. So, we know that current is basically per unit at a particular unit time, how many electron has flowed through a particular point, right. So, if you know that this is entity A, this is entity B, and you keep a watch somewhere in between A and B, and you say, okay, this many electron has passed at that per unit time. So, I can measure the current, which is essentially denoted as ampere, and the voltage change denoted as volt, right. So, the whole electricity, the whole field of electricity, electrodynamics, whatever you want to, how far you want it to go, is all about this transport of electrons from one point to another point, and you need tools to measure that. So, you have to have a background and like how you do all these measurements. So, and one more thing I will add, electrochemistry is a surface science. It is only at the surface. It is not really getting the core of the material. It is the electron transport which is happening at the surface. So, if you look at biology now, so the course title is Bio-electrochemistry. So, the biological system among proteins, there are always transfer of electrons from one protein to another. If you look at the whole mitochondrial electron transport chain, you will see that there are different electron donation or electron accepting entities which are sitting along that membrane. Similarly, if you look at the chloroplast, same way, or forget about it, if you do not even look at them, look at the different proteins they are interacting, there are always transfer of electrons. So, electron transfer is the key point which dictates several reactions, most of the reactions, and is essentially could be used as a tool to quantify the kind of reaction or qualify the kind of reactions. So, in electrochemistry, we deal with measuring this different change in voltage and current in the form of voltammetry, amperometry, chronoamperometry, a series of techniques. So, in this course what we will essentially will be doing, we will talk about the very basic fundamentals which a biologist can use, the basic fundamental electrochemical tools which in biological context could be used to develop sensors, to understand some of the most fundamentally beautiful reactions which are taking place inside the cell or in nature, as well as in waste remediation and several other, like starting from fuel cell to batteries to supercapacitors, bio-inspired designs, series of them. So, in order to appreciate all these things, one need to have a very clear conceptualization of electrochemistry. Then one can apply all these things in the biological setup. So, this course is fundamentally targeted to the biologist who have inclination to understand electrochemistry and use it as their research tool. So, I welcome you all for this journey into the world of electrochemistry from the biological perspective. Welcome you all. Thank you.